If there's one idea that makes modern medicine modern, it's germ theory. The idea that specific pathogens like bacteria or viruses can cause specific diseases. Antibiotics, vaccines, mosquito nets, they're all applications of germ theory. As someone who makes videos about medical history, I spend a lot of time reading about germ theory. But before this video, I'd never done a proper deep dive into its history. I knew Louis Pasteur was involved and that his experiments on spontaneous generation led to pasteurization. I was also familiar with the work of Robert Koch, who discovered the causative germs of anthrax and tuberculosis. And I knew these two had personal beef, so I was looking forward to reading about an old school scientific feud. But telling that story would still just be your classic great man history, and those aren't the most exciting stories for me. Like, germ theory brought us from bloodletting and mercury injections to antibiotics in less than 100 years. Now that's a story I'm interested in. So I had a few research questions for this video. What did Pasteur and Koch actually do? What kind of pushback did germ theory have to overcome? And how does germ theory hold up to scrutiny in modern day? To answer those questions, we need to understand how people thought about disease in the year 1800. Because your average non-doctor believed in some variation of contagion theory, this idea that disease could spread from person to person. Scholars like Claudius Galen in ancient Rome and Girolamo Fracastoro in 16th century Italy wrote about this a bunch. They thought disease passed from person to person through little seeds, or what they called semina. But in reality, people didn't really care what old academics thought. Contagion just made sense. Like, if your roommate got tuberculosis, you'd probably start to worry that you'd catch tuberculosis too. It was scientists that were harder to convince. They accepted that some diseases like syphilis or rabies were contagious, but epidemic diseases like cholera or bubonic plague or leprosy were more questionable. Like, someone could get the plague who'd never met another plague victim. So scientists were more likely to believe in traditional miasma theory, the idea that bad air caused disease. I got a video all about its history if you want to check it out. But by the mid-1800s, there was a bunch of evidence saying that miasma theory didn't really work, but scientists didn't have anything better to replace it with. They still had no better way of explaining how disease could move over a large distance, or how it could infect a lot of separate people at the same time. Plus, contagionists still didn't know what the contagious thing was. It could have been a poison that putrefied in the body, or it could have been an animalcule that you could see under a microscope. There was also this new model called zymotic theory. This idea said that disease came from little things called zymes that were even smaller than cells and under the right circumstances morphed into bacteria. Nobody had great evidence for any of their theories, but by the 1830s, things were starting to change that would make gathering that evidence much easier. The first was that doctors were finally starting to shift away from the old school humoral theory of disease. So instead of thinking about illness as an imbalance of the four bodily humors, blood, phlegm, black bile, and yellow bile, they started looking for issues in individual pieces of anatomy. And to do that, they did autopsies. They'd record someone's signs and symptoms when they were alive, diagnose them with whatever disease, and then when they died, they cut them open and looked for any patterns in the anatomy. Like, if a patient had breathing issues when they were alive, then doctors would look for anything weird in the lungs on autopsy. This became known as the field of pathology, and it pushed doctors to go back to basics and ask, what is and where is disease? I got a video all about that one too. The second big thing was cell theory. By 1837, two scientists published their papers on this new idea that every living thing was made up of cells. One of them even helped discover that yeast, like the ones involved in fermentation, were living microbes themselves. And when combined with the new field of pathology, cell theory prompted doctors to start looking for problems at the cellular level, what's called cellular pathology. So to summarize, we understood disease as a problem with individual body parts, we had the technology to look at that body part in great detail, and oh, by the way, everything you see under the microscope is alive. It's no wonder scientists were obsessed with microbes at this moment in history. One of those scientists who thought up of cell theory, Theodor Schwann, was lab mates with an anatomist named Jacob Henle, who anatomy students might recognize from the loop of Henle in the kidney. And fun fact, they were also roommates.
Oh my god, they were roommates.
Henle eventually branched out from anatomy and started thinking about disease transmission. And in 1840, Henle came out with an essay where he asked how it was possible that lots of people in the same area could get sick with the same thing. Henle thought that some diseases were spread by miasmas, some were contagious, and some were a hybrid miasmatic contagious. But he thought that scientists had been missing a nuanced distinction throughout the years. That those seeds of disease that Galen and Fracastoro talked about weren't miniature versions of disease, they were microscopic living things that caused disease. Likewise, bad air didn't cause disease, but air could carry things that caused disease. Unfortunately, Henle thought it would be impossible to prove. He wrote, Showing germs really are the effective cause could only then be empirically proven if one were able to isolate both the germs and fluids of Animalcula and the organisms and fluids of Contagia and then observe the effect of each of these separately, an experiment we will probably have to do without. Then he proposed 3 rules, or postulates, that a microbe needed to meet in order to qualify as the cause of a disease. The microorganism had to consistently be found in bodies suffering from the disease. The microbe needed to be isolated, then when that microbe was put inside a new healthy host, it had to produce the same disease. It was a sound idea, but there were some issues with the execution. Like, if you wanted to study tuberculosis in mice, you'd take some mice with TB, let them get sick and die, look for some kind of infected-looking material in their bodies, then put that material into healthy mice hoping that they develop TB. Then, when that batch of newly infected mice died, you should be able to pull the tuberculosis germ out of them, too. But the mice didn't always die, which made confirming that last step really tricky. So Henle was stuck. Luckily, one of his students would come up with a solution. But I want to build some suspense. So to recap, by the mid-1800s, the mainstream scientific thought was all over the place. It was a combination of miasma theory, zymotic theory, and contagionism for certain diseases. And that's where Louis Pasteur comes in. Pasteur grew up in the French winemaking region of Arbois, and wasn't a particularly great student growing up. He eventually earns a science degree, then gets a job as a physics professor before becoming a graduate assistant in a chemistry department. And that's where he gets his first bit of notoriety. Pasteur's breakout discovery was the observation that some crystals had distinct mirror opposite shapes of themselves, a phenomenon called chirality, and it's a big deal in modern chemistry. After that discovery, Pasteur started getting a little bit more respect for his research, so he spent the next decade studying crystals. Then in 1854, Pasteur got a job at the University of Lille, where he started studying the process of fermentation. Because for as long as humans had been drinking fermented beverages and eating fermented food, we didn't know how fermentation worked yet. Some scientists thought it was entirely a chemical process, while others thought it was more of a biological thing. By the late 1700s, a few scientists noticed that yeast cells tended to show up in fermented foods and alcohols, making it seem more like a biological thing. And like I mentioned in the miasma video, scientists couldn't ignore how similar fermentation was to putrefaction, the process of bloating and rotting that happens to a corpse after it dies. Pasteur was a chemistry guy, so at first he thought fermentation was mostly chemical, but after looking at a bunch of fermented alcohols under a microscope, he saw microscopic critters everywhere, which changed his mind and made him think it was a biological thing. Pasteur published a paper in 1857 showing that milk spoiled because microscopic yeast fermented the sugars in the milk into lactic acid. And in 1858, he proposed that yeast process sugar into alcohol. He still didn't have a super strong understanding of how it worked biochemically, but he saw so many microbes in the sour milk and alcoholic beverages that he figured they had to be responsible for fermentation. Like I said, plenty of other scientists had proposed that fermentation was a biological process, so Pasteur's research wasn't groundbreaking. But it did make Pasteur wonder whether fermentation was involved in disease, too. But before he could tackle that question, he'd need to deal with some very public drama. This is Antoine Béchat, a French scientist who had a big problem with Pasteur. See, Béchat published a similar hypothesis on fermentation in 1855, 2 years before Pasteur's paper, but he interpreted the results through the lens of zymotic theory. Béchat studied how cane sugar turned into glucose and fructose in water, and claimed that they broke apart thanks to a substance called zymase, which would have made fermentation more of a chemical process than a biological process. But after Pasteur became a celebrity with his first paper, Béchat started saying that he forgot to mention there was a mold growing in one of his solutions, which meant that he had shown fermentation was a biological phenomena before Pasteur did. And oh, by the way, Pasteur had actually stolen his idea. Pasteur responded by calling out the timeline. You can't go back in time and tweak your conclusions to fit new evidence. This was the first time of many that these two had beef throughout their careers. Whenever Pasteur got attention for a discovery, Béchat was there to try and debunk him. Pasteur moved on from fermentation and started studying the phenomenon of spontaneous generation, a super old idea that living things come from non-living things. This is when he did the experiment you probably learned about in middle school science class. In 1860, Pasteur had these special flasks made and filled them with a broth that was really good at growing bacteria. After heating them to kill off any existing bacteria, he either left them open and exposed to air, or closed off, which prevented air from getting inside. At the end of the experiment, the closed containers didn't have any bacterial growth, while the flasks exposed to air had plenty. Since bacteria didn't in fact grow in the closed containers, he concluded that spontaneous generation wasn't really a thing. Something from the air must have gotten in the open flask and contaminated it. Just like his work on fermentation, this experiment wasn't totally original either. An Italian scientist named Francesco Redi did something similar with meat and jars back in the 1600s. The difference was the scientific environment around each scientist. Redy's experiment was the first of its kind, and he didn't really know how to explain his observations. But by the time Pasteur did it, scientists were more familiar with microbes and open to debunking this ancient idea of spontaneous generation. This experiment wasn't important because it was the first one. It was important because Pasteur got people to listen to him, and because it led to a very cool application. He noticed that when microorganisms grew too much in beer or wine, the drink would spoil. So he came up with a technique for killing off those bacteria that involved heating up the beverage to somewhere between 60 and 100 Celsius. He published his findings in 1863 and patented the process in 1865. And today we know it as pasteurization. This was a big deal for France's economy, since pasteurization allowed the French to export more wine. But of course, this is a medical history channel, so we gotta talk about the medical applications. Because some of the people reading Pasteur's work were doctors who started applying his ideas to real-world medicine. Most notably, a surgeon in Scotland named Joseph Lister read Pasteur's work on fermentation and thought it might help him in the surgical ward. Recent advancements in surgery, like anesthesia in the 1840s, made surgery more accessible, but way too many patients were dying from infections after surgery. After reading Pasteur's work, Lister reasoned that if putrefaction and fermentation started when a substance was exposed to air, then sanitizing the air around an open wound should prevent infection. And his air sanitizer of choice was just so miasma-inspired. He'd heard of a town in Germany that sprayed this stuff called creosote on their sewage to keep down the bad smells. And since everyone thought the smells associated with garbage were the same as the ones in putrefaction and infection, Lister thought this trash deodorant might work against infection too. Creosote is a byproduct of tar manufacture, so he tried a bunch of other tar derivatives before arriving at phenol, or carbolic acid. When he sprayed this stuff on his surgical patients, death from post-op infection fell from 45% to 15%. This became known as antiseptic surgery. I got a video all about it that you can find here. Antiseptic surgery was a cool application of this thing that was becoming known as germ theory, but scientists still didn't really know what caused disease. So let's go back to France. Between 1855 and 1865, there was this big outbreak of pepper disease, or pébrine, in France's silkworm population. The worms developed these little brown spots and lost their ability to spin silk, which was important because France exported a lot of silk. In 1865, a few scientists started trying to figure out what was causing the epidemic, including Pasteur, who had been commissioned by the French government, and Béchat, who funded his own research. And of course, according to Béchat's own reports, he figured it out right away. He claimed the disease was caused by a zyme called pébrine that lived on the outside of the worm's eggs, and a quick spray with carbolic acid ought to fix the problem. Pasteur thought Béchat was full of crap. He thought there was no way there was a germ on the outside of the egg. He's quoted as saying, "I should be as happy as those persons who, like Béchat, acquire ideas without the need to first accumulate facts." It's a sick insult, but Pasteur hadn't done any better. He thought maybe the disease behaved kind of like cancer, or maybe tuberculosis. He didn't know for sure, but he was certain Béchat was wrong. But after 2 years of making little to no progress, a few German colleagues convinced Pasteur that Béchat was onto something. In an 1867 letter to the Secretary of the Agricultural Committee, he admits, "Poor Monsieur Béchat is at this moment one of the most curious examples of the influence of preconceived ideas gradually turning into fixed ideas. All his statements are so biased that I wonder if he has ever observed more than 10 silkworms in his life." Clearly, Pasteur was a very humble guy. Now, microzymes aren't a real thing, but Béchat's idea that something was attacking the eggs from the outside was likely correct. Unfortunately, his attempts to stop the disease by spraying the silkworms with carbolic acid weren't working, so Pasteur came up with a new idea. He thought that if you separated the diseased silkworm cocoons from the healthy ones, you might be able to at least stop the spread of disease. Luckily, it worked. So Pasteur got credit as the hero since his invention once again saved the French economy. Béchat, on the other hand, never saw the spotlight again. But put a pin in that, because Béchat shows up towards the end of the story. Pasteur thought these diseases might have been caused by microbes that putrefied in worms' bodies, kind of like how we saw microbes ferment wine. He ended up isolating 2 different types of bacteria and found that each one was found in a different disease. And this turned out to be one of the most important insights in germ theory, because Pasteur was showing disease specificity, the idea that a specific disease is associated with a specific bacteria. But that was an association. Scientists still hadn't shown the cause and effect relationship between germs and disease yet. And Pasteur would start to make that connection thanks to an international rivalry with a German scientist named Robert Koch. Also, his name is properly pronounced Koch, but that one pronunciation took me like 5 tries, and if I'm gonna get through this script in one sitting, I need to Americanize it. Sorry to the German speakers out there. Robert Koch was 20 years younger than Pasteur, and a much better student. In 1862, when he was 19 years old, Koch started at the University of Göttingen, and after a few semesters started working for a scientist we've mentioned already, Jacob Henle. The guy who said, here's how you would prove a germ caused disease, but it's impossible. After graduating, Koch moved out to rural Germany and started studying anthrax, a bacterial infection that was wrecking local livestock populations. And this research would turn into the experiment that Henle said was impossible. Earlier researchers had already identified Bacillus anthracis as a potential cause of anthrax, so Koch knew what to look for in the animals. He took samples from the spleens of cows and sheep that had died from anthrax, gave it to lab mice, and watched them develop anthrax. And here's the innovative part. Henle assumed he'd only be able to take samples of bacteria from the infected animal after they'd died, but Koch came up with a way to extract tissue from the animals while they were still alive. And since they were still alive, the bacteria could keep multiplying, which gave him an endless supply of uncontaminated Bacillus anthracis to give to mice. Then he'd take that bacilli and culture it, meaning he let the bacilli reproduce in some kind of medium. He very cleverly used the goop inside ox's eyes for this experiment. Then he'd take that pure culture, inject it into healthy animals, and watched as they develop anthrax. Then he'd extract bacilli from the newly infected animal, put that on a slide, dye it to make it more visible, and looked at it under the microscope. And this was a big frickin' deal. These experiments with anthrax were the first time that someone had isolated a microorganism and caused disease with it. Remember specificity. These animals didn't develop cholera or syphilis, they were given Bacillus anthracis and developed anthrax. This was the start of modern germ theory. In 1876, Koch published The Etiology of Anthrax Disease based on the developmental history of Bacillus anthracis. And in 1877, he published on anthrax again, this time describing the bacillus in even more detail. He even included a photo of it, one of the first photos of a bacterium ever. In an 1878 paper, he says, you should be able to repeat the same process for any disease-germ combo and find the cause of disease. So just like his mentor did in 1840, Koch outlined step-by-step instructions to show causation. You'd have to find the germ in all subjects with the disease, then when you put that germ into a new animal, the animal would have to get the same disease, and each disease would be caused by a distinct germ. This process would become known as Koch's postulates, sometimes Henle-Koch's postulates to acknowledge the mentorship. Koch ran into some issues with the postulates over the years, which we're going to talk about at the end of the video, but by 1878, he had a good method for identifying causative agents of disease. At this point, Louis Pasteur decides to get involved with his own anthrax research. And at first, he mostly copy-pasted Koch's work and claimed it had never been done before, which at this point, you probably expect from Pasteur, but this was pretty light considering what was about to come. Because as soon as Koch published his anthrax work, Pasteur started working on an anthrax vaccine. See, Pasteur had successfully made a vaccine against chicken cholera a few years earlier with a technique called attenuation, which involves creating a weakened version of the germ, then introducing that attenuated germ to your immune system, letting you build immunity to the pathogen without getting sick. Some modern vaccines are live attenuated vaccines, but it was brand new back then. Pasteur found that the bacteria that causes chicken cholera got weaker after just letting it replicate a bunch. But that same technique didn't work to attenuate the anthrax bacillus. So he and his team tried using heat, and found that the germ lost its ability to make its host sick when it was heated between 42 and 43 Celsius. We'd say that's a less virulent form of the germ. This became their vaccine candidate. Always the showman, Pasteur did a very public experiment in 1881 where he successfully vaccinated a bunch of sheep against anthrax. Successful in the fact that the sheep that were vaccinated lived and he got a bunch of media attention for it. At this point in history, Germany and France hate each other, so Koch and Pasteur's rivalry takes on this international competition. And after the anthrax vaccine demo, Koch accuses Pasteur of cherry picking. But this kind of goes past a good faith critique of the science and gets a little too ad hominem in my opinion. Pasteur follows the tactic of communicating only favorable aspects of his experiments, and of ignoring even decisive unfavorable results. Such behavior may be appropriate for commercial advertising, but in science it must be totally rejected. At the beginning of his Geneva lecture, Pasteur placed the words, "Nous avons tous une passion supérieure, la passion des vérités." We have no passion greater than the passion for truth. Pasteur's tactics cannot be reconciled with these words. His behavior is simply unexplicable. Koch was not messing around. Koch moved into a new and improved lab by 1880, and at that point, it was game on. In 1882, Koch discovered Mycobacterium tuberculosis, the bacteria that causes TB. And that same year, Pasteur came up with a vaccine for rabies, making it the first human vaccine created with germ theory. You bet I'll have a video about that one. In 1883, Koch went off to Egypt to study cholera, and using his postulates, he attempted to isolate Vibrio cholerae, the bacteria that causes cholera. He'd moved on from culturing bacteria in cow eyeball fluid and tried using nutrient-dense broths. Unfortunately, this media wasn't suited for isolating bacteria since everything grows in broth, which made finding the germ of interest quite the challenge. Gelatin seemed like a promising alternative, but certain microbes broke it down too easily, and it melted at way too low of a temperature to be useful. But one day, one of the lab assistant's wives, Fanny Hess, had an idea. She'd been making vegetable jellies with this substance that fit all the criteria the lab was looking for. It was clear, it had a high melting point, and it turned out it didn't get broken down by bacteria. This substance was called agar, and the Koch lab started using it right away to culture isolated species of bacteria. A super useful technique when you're trying to identify the single microbe that causes the disease. Then another assistant designed a circular glass dish that they could use to contain the agar. The assistant's name was Julius Richard Petri, giving us the Petri dish. And this was another huge game changer. The combination of Petri dish plus agar let scientists culture tons of new types of bacteria and are still in use today. So using pure cultures, Koch could ask whether Vibrio cholerae actually caused cholera or whether it just showed up in the intestines during cholera. Unfortunately for Koch, the bacteria only causes the disease in humans, so he wasn't able to test in animals, but he still was able to show that cholera never happened without the cholera bacteria. And that brings us to one of the big modern misconceptions around germ theory, that Koch's postulates are strict rules that need to be followed to show causation. Koch himself never considered his postulates to be hard criteria for determining cause and effect of disease. They were a useful starting point, and sometimes worked perfectly, but Koch himself didn't see them as gospel. In fact, in 1884, Koch published a paper about tuberculosis where he modified his postulates into the version that most biologists know today. Points 1 through 3 are basically repeats of his original postulates. There must be a germ in every case of the disease. That germ must be a unique living microbe that can be distinguished from other microbes, and there must be a logical mechanism for the germs to cause the disease. Points 4 and 5 came from this new tuberculosis research. You must be able to isolate and culture this germ outside of the test animal, and finally, when you put that germ into a new animal, that new animal has to develop the same symptoms as the original animal. Again, this wasn't a prescription for how to find new germs, but it did work for TB. And while there were definitely holdouts, which I'll talk about in the next section, this is when other medical professionals start accepting germ theory. Okay, so while Koch was working on TB and cholera, Louis was getting ready to go institutional. He and his team opened the Pasteur Institute in 1888, and after that, scientists there found everything. By the turn of the century, scientists had used germ theory to identify the causative agents of gonorrhea, then plague, then dysentery, tetanus, and staph infections, many of which happened thanks to the scientists at the Pasteur Institutes. Pasteur himself died in 1895 and was buried at the Pasteur Institute in Paris. Koch remained a living legend in the scientific community, until one high-profile professional embarrassment that I talk about in this video. Now, in general, the medical profession starts accepting germ theory in the 1880s and applying it in the 1890s. By the time of Robert Koch's death in 1910, most serious scientists and physicians were on board with germ theory. But like with any revolution, there were holdouts. Remember that French scientist who hated Pasteur, Antoine Béchat? He proposed an alternative to germ theory called terrain theory, which said our bodies served as homes, or terrain for zymes, and when we're unhealthy, the terrain becomes easier for those zymes to turn into bacteria and cause disease. Béchat didn't see disease as a battle between perfectly sterile bodies and dirty bacteria. He thought of it as more like an imbalance. Béchat died in 1908, after which some of his followers posthumously published his final work in the 1911 book, The Blood and Its Third Element. I read this thing, and he wasn't exactly subtle with his goal. His main hypothesis was that he was right all along and that Pasteur was both wrong and also plagiarized Béchat's correct ideas, which don't seem like compatible accusations. He starts off with a bang, comparing himself to Galileo. You know, he's being silenced by the system and the scientific community was out to get him. Then he describes how everything scientists know about blood coagulation is all wrong. He claims that the component of blood that clumps blood cells together into clots, what's called fibrin, is actually a, quote, false membrane of microzymes. Therefore, zymotic terrain theory is really the one true theory of disease, and germ theory is wrong, and Pasteur should be disgraced. Obviously, there were some big jumps in logic there, but I think even fans of Béchat recognize that this book isn't about communicating his science, it's about his reputation. Men will literally invent new models of disease instead of going to therapy. This book is where the trouble started. Antoine Béchat kicked off the germ theory denialism that's still around to this day. Béchat's followers were obsessed with him. The preface to the 1911 1911 translation of the book refers to him as an all-caps master and says the master's arch-plagiarist Pasteur is dead, though his evil work lives. I would like to point out, though, that Béchat refers to this evil work, germ theory, as the greatest scientific silliness of this age, which, come on, that's adorable. A few years later, in 1923, a woman named Ethel Douglas Hume published Béchat or Pasteur, a lost chapter in the history of biology. And it's another agenda first, science second kind of a book. I have links to this one and all my sources in the description. Most of the book is about how Pasteur plagiarized everything and Béchat had been silenced his whole career, but really, all of this is a setup so that the author can go on an anti-vax rant saying that vaccines are the worst tyrannies imaginable and should be resisted even to the death of the official who is enforcing it. So yeah, she had some strong opinions. The rest of the book covers the usual anti-vax talking points, but my favorite part was the line where she recommends that everyone adopts a raw vegan diet and it'll be literally impossible to get sick again. This book was essentially Goop before Gwyneth, and the reviews from this time are just incredible. The scientific journal Nature reviewed it in 1924 and said, "This book is an attempt, not the first in recent years, to resuscitate the name of Béchat and his microzymus theory, which was presumed to have been utterly demolished by the labors of Pasteur. The work is replete with extravagant language on the virtues of Béchat and a tirade of the defects, if not vices, of Pasteur as an investigator. Béchat is described as a rare luminary of science, a supreme benefactor of humanity. We are told that he stood on an ethical plane above his fellows, that truth, not self, was Béchat's lodestar, and he is compared to Galileo. The solid fact remains that most of his work has been discredited as inaccurate, and although he wrote an immense amount, he plunged deeper and deeper into error. However high the opinion of the author is on the virtues of Béchat, he has utilized a fair part of the book to exploit his own antimicrobic and anti-vaccination views." Basically, even the review was like, yeah, this is thinly veiled anti-vax propaganda. And just like a lot of other 19th, early 20th century conspiracy theories, there are still people who believe that Béchat was right all along and we've got microzymes in all of our cells. Just like Hume in 1923, they tend to be into alternative medicine and not into vaccines. But here's the thing, even without Béchat's followers, actual scientists have critiqued and updated germ theory. I mean, it would be bananas if the theory hadn't been refined since 1885. And to come back to the Galileo comparison, it's kind of like how the idea of heliocentrism has been updated and refined to reflect new discoveries in astronomy and physics. The biggest critiques revolve around the consistency of Koch's postulates. And so, in our little conclusion here, let's ask, how have Koch's postulates stood the test of time? After his famous tuberculosis paper, Koch started to notice that the same germ could cause different diseases by different mechanisms. Like, it might give off two different toxins that cause different symptoms, which muddled up postulate number 3. There was also the issue of asymptomatic carriers, people who had the disease in their bodies but didn't show symptoms. And later on in his career, he was never able to get test animals sick with certain diseases like typhoid fever or leprosy, despite giving them the germ, and that violates postulate number 5. And just before Koch died, a researcher at the University of Chicago found a new genus of microorganism that consistently showed up in cases of Rocky Mountain spotted fever. This weird new microbe fulfilled every one of Koch's postulates, except for number 4. It couldn't be cultured in a petri dish. But the researcher found the microbe in so many cases of the disease that he was still pretty sure he'd found a cause and effect situation. He ended up presenting his findings with an appropriate mixture of confidence and caution. In his 1909 paper in JAMA, he says, "In spite of the constancy with which these bodies were found," talking about the germs, "it did not seem justifiable to claim that they represent the microparasite of the disease for 2 reasons. 1, the very complex morphology of the blood, especially in febrile states, when various cells and probably their nuclei are subject to unusual disintegration. 2, because of my inability to cultivate a microorganism of this character from infected blood by the use of ordinary and some unusual culture media, under various conditions of cultivation, or by other means to obtain it in satisfactory condition. Basically, the blood made it hard to see, and I didn't meet postulate number 4, so maybe it's not cause and effect." Luckily, future research would confirm that this germ genus didn't only cause spotted fever, but epidemic typhus as well. This genus of germ was named Rickettsia, after the Chicago researcher who discovered it, Howard Taylor Ricketts. Okay, so Koch was starting to find exceptions in his postulates, and now other researchers were too. But the biggest issue focused on a class of germs nobody had actually seen at that point: viruses. In 1884, one of Pasteur's assistants, Charles Chamberlain, came up with a device that could filter bacteria from a solution which left anything smaller behind. Experiments showed that even when you filtered out the bacteria, the solution could still make experimental animals sick, so there must be something smaller than bacteria that caused disease, which scientists identified as either toxins or viruses. They didn't know anything about the biology of viruses yet, just that there was something physically smaller than bacteria that could cause disease. And by the 1920s, a few scientists proposed the modern idea that viruses actually replicated inside cells. Cool, but this violated Koch's postulates because it meant that viruses couldn't be cultured in agar. They'd need to be cultured in living tissue samples. And that took until 1931, when scientists at Vanderbilt University found an appropriate type of tissue to culture a specific virus. That same year, researchers discovered that influenza was caused by a virus, not a bacteria like they'd thought for decades. I made a video all about how that story influenced vaccine development during the 1918 flu pandemic. You can find it here, and I swear I'm done plugging videos. In 1937, a scientist at the Rockefeller Institute named Thomas Rivers published a paper titled Viruses and Koch's Postulates. In it, he says, with the development of the science of immunology, however, immunological reactions added much to the knowledge of the specific relation of microbes to disease, and now it is possible to bring excellent evidence that an organism is the cause of a malady without the complete satisfaction of the postulates. Basically, we can say that a microorganism causes disease without checking every item off of Koch's postulates. By the 1940s, a bunch of new technologies like electron microscopes became a thing, which allowed scientists to identify viruses faster than ever before. And in 1957, Dr. Robert Hubner published The Virologist's Dilemma and explained that all this new detection technology was actually a problem. Virologists were finding novel viruses faster than they could study them. Furthermore, there were a bunch of viruses that showed up in both sick people and healthy people. So Hubner proposed that in addition to lab work, viral diseases would need epidemiological evidence too, at least if they wanted to show that a virus caused a specific disease. And research published in the 60s documented how certain acute respiratory diseases behaved even weirder. They found that the same clinical syndrome, the same presentation of signs and symptoms, could be caused by multiple viruses. And that's problematic because different viruses may respond to different treatments. And if you're choosing treatments based on symptoms, you might pick the right treatment for the wrong germ. And lab work doesn't necessarily help the diagnosis either. Depending on the type of illness, scientists couldn't find a causative germ in anywhere from 25 to 75% of cases. And then there was the big question of the 80s and 90s. What causes this new thing, acquired immunodeficiency syndrome, or AIDS? Scientists first isolated the human immunodeficiency virus, or HIV, in the 80s, but couldn't find it in anywhere from 20 to 50% of AIDS patients, which violates postulate number 1. In the late 80s, a scientist named Peter Duesberg used that as evidence to support his hypothesis that HIV didn't cause AIDS. He blamed AIDS on recreational drug use and antiretroviral medication, like AZT, which was one of the only AIDS medications available at the time. Needless to say, that hypothesis didn't age well. Then in 1988, a trio of well-respected scientists at the National Cancer Institute responded in the journal Science saying that HIV did cause AIDS, regardless of Koch's postulates. They say the Henle-Koch postulates of 1840 and 1890 were formulated before the discovery of viruses. They are a useful historical reference point, but were not regarded as rigid criteria by Koch himself and should not be regarded today. And these days, the scientific consensus is that HIV does cause AIDS, which is based on the totality of scientific evidence to date. And that gets to the take-home message of this video. Science is a process, and the collective knowledge that science builds grows over time. So we can't think of germ theory as right or wrong. We need a more nuanced view. I think of germ theory more like a foundation that biology builds on. Like we learned that viruses can cause disease. All right, let's refine our criteria. We noticed that chronic diseases couldn't be explained by the same criteria as infectious diseases. Cool, now we have a better idea of what germ theory does and doesn't apply to. We learned that the microbiome is a thing and that not all bacteria cause disease. Cool, let's explore that more too. But the main idea that a disease doesn't happen without the causative germ is still useful and accepted by infectious disease researchers. The virus that causes influenza might not fulfill Koch's postulates, but my annual flu vaccine keeps me from getting the flu because it trains my body to recognize the influenza virus. Rivers encapsulates my feelings on this when, in 1937, he said, "The cause of viral diseases is known, and that Koch's postulates as proposed by him do not have to be fulfilled in order to prove that a virus is the cause of a disease. However, the spirit of his rules of proof still holds, in that a worker must demonstrate that a virus is not only associated with a disease, but that it is actually the cause. The methods for doing this are different from the ones used by Koch, but are equally efficient." Germ theory itself was a massive academic achievement, but the practical applications had a bit of a lag. Doctors in the year 1900 knew the cause of so many diseases and could prevent them fairly well with vaccines, but they didn't have the treatments to fight them once someone fell ill. For that, they would need the biggest therapeutic development in the history of medicine: antibiotics. Check out this video to keep learning.